So thanks again for all our speakers and panelists today. Let's see. My name is David Gerhold. I work at NIH at the National Center for Advancing Translational Sciences. I've been charged with making a few closing remarks. So I've tried to extract a few lessons from what we've gone over today. So first, I'm thrilled and honored to be here and considering these problems with you. A few words about NCATS. We're part of the TOX 21 consortium. Our central expertise is quantitative high throughput screening, so we do a lot of the screening for TOX 21. And then what I do is to work on validating models for dopaminergic neurons, for endothelial cells, and a few other cell types. I also do work on high throughput gene expression profiling and genomics. Let's see. Today, we talked about tools and applications of those tools. And so tomorrow, what we're going to cover is some additional brainstorming as to what we can do with these technologies. And we'll get the guidance of some of our experts who have already worked with CRISPR quite a bit. And we'll also consider how to use genome engineering to make health decisions. And so this is something that that I think requires some thought and will benefit from some thought and some discussion in advance. Let's see. So these are the two panels which we'll go through tomorrow. Let's see. The first one is about genome editing tools. And in what ways could they inform environmental health decisions? The second one is perspectives on use of this technology for environmental health decisions. Okay. I wanted to say a few words too about the workflow process and how much work things are. So first, I'm still a novice at using CRISPR, and we have a few projects going. But immediately, the CRISPR allows us to make to address the region of interest and to make cuts, and that worked right off the bat and is fast and easy, and so that part is wonderful. So the bottleneck is no longer addressing the region of the genome that we were interested in, as it used to be. The bottlenecks for most of these projects are now number one, can I introduce DNA effectively and efficiently to the cell type of interest? And we had some discussions in the meeting and in the back of the room about how we can address primary cells. And so I think the lentiviral vectors and maybe the AAV vectors will even allow us to address primary cells with some of these approaches. The second part of the workflow is the need to grow colonies from single cells. And so what we're doing, in fact, what someone in my lab is doing today is doing cell sorting on electroporated neuronal cells, and then encouraging those single cells to grow into colonies. And so for people working with stem cells, for example, that's probably the limiting factor is we can transfect them or electroporate them fairly efficiently, but you still need to get them to grow into colonies from single cells, and that can be done, but it's a fairly inefficient process. So the second thing to consider. And the third part is simply growing those colonies, doing a lot of cell culture, and doing PCR to identify the mutants you're interested in, PCR and sequencing. And so that's the third part of the workflow that persists in, I think, all of these approaches. Okay. So the slide I wanted to show you here is my simple-minded way of brainstorming for how to solve problems. And so what I do occasionally is I list problems that I'd like to solve, which are goals on the right-hand side of this slide. And then I list techniques, some of my favorite tools in genomics or in cell culture. And then I pair them up in pairwise comparisons, and I say, can this tool help me solve this problem, or this problem, or this problem? No, I go on to the next tool, and I pair that with each of the problems I want to solve. And so that's my simple brainstorming approach. And so I made this slide to help people think about what problems they want to solve. And so you should add the problems that you want to solve to this. And then the techniques that are available to us using CRISPR, and then you can look to see how you can solve those problems. And if I look down at the, let's see, 1, 2, 3, 4, 5, 6, the 7th bullet here, sorry, the 8th bullet, is to select or identify important genes from engineered cell populations. And so we've seen some wonderful examples from Chris Volpe and from Liuping Zhang. And so this has been demonstrated with transcriptional activators or repressors, with gene knockouts, and it could potentially be done with any of these first 6 bullets, including RNA knockdown by RNA cleavage, changes in DNA methylation, transgenes, or mutation introduction correction. And then finally, it's been pointed out that we can make large genomic deletions. There's a physical process by which we can label genomic loci, similar to the FISH, the fluorescence in situ hybridization technique. And Cas9 has also been used for physical selection of genomic loci to enrich for mRNAs that you want to study, for example. And so this is the list of the tools that I've been able to come up with for CRISPR. Can be applied to a variety of cell types, and we've discussed that at some length. And then you can make a list of your own goals and brainstorm for tomorrow and think about what you'd like to do with the technology. So thank you, and I think some people are going to dinner this evening. Is Keegan or somebody organizing? Okay, so thanks for your attention.